Hello, everyone. Welcome.
Hearing, sorry.
Marcella. Not this one. Okay, do you see the slides?
Yes.
Somehow my screen is covering you guys, so I can't see you. All right, so let's begin. Um, so today I'm gonna talk about the last step of the pipeline, the freebase polishing, and what we have to do before sending for curation. And this time we have our special guest, Kirsten from Sanger Group, who is leading the curation team that is taking care of all our genomes at the end. We'll talk about the last part here. We'll tell what the specific, what's happening in between the scenes in this step, and yeah, which is the last step. It's currently in the investigating pipeline folder, the freebase polish step. But before you begin any process, please make sure that you're using the latest workflow in the VGP Tools project. Just check whether you're using the latest one. Chai made a new scheme in the workflow names, so all the workflows comes now with a date in there. So the dates, take whatever is the latest one. Okay, so yeah, the workflow. So the intent for this step is to correct base pair errors based on the 10x read alignments. We're doing this in two steps. We do long range alignment. Just as a note who might be familiar with 10x Long Ranger, we're not using Long Ranger WGS, which does the variant calling inside. They have a limited number of reference IDs that can be processed to do the SV calling and phasing, which breaks our pipeline. So we are just using the alignment part, which does the alignment and refines the alignment based on the 10x barcodes. And then we use freebase to call the variants. Here we are assuming, since we are aligning 10x Illumina reads to the same individual, all the variants you get called here are base pair errors. And using those, we do back, which is the consensus step. And Julio made the QV app, which gives us an estimate of the QV. So this whole process here is done twice. And at the end, the assembly stats app is optimized the final assembly to the primary and alternate sets. So here are done automatically. There are some special parameter sets. For freebase, after a few evaluation, we decided the latest version, which has this new feature, saying it's called skip coverage, where we are taking the mean coverage estimate from the 10x long ranger alignment and anything above 12 times the mean, we are skipping to call variant calls, because this is obviously a collapsed region or a highly repetitive region, which is difficult to make a true assumption for building the consensus anyway. So the reason for putting this in here was because of the time constraints we have on DNA Nexus. We can't just let Freebase run over a week. So that's the biggest motivation to put this in here. And for the consensus, we use a pretty loose quality filter and take the alternate allele, homozygous, when it's called, when it has more than 2 heterozygous alleles, especially these happens for indel calls, we take the major allele that has the most support. And the QV calculation, I've got asked how we do this calculation several times. It's basically using the same scheme for getting the Phred score. So essentially it's measuring, given how many base pairs we see in our consensus, how many base pairs are errors or affected in our case. So here we are measuring the QV stats for the assembly that's gone this polishing step. So to do the correct estimate, in theory, we have to run this whole pipeline 3 times and collect the Freebase calls or the number of base pairs that Freebase suggests to be erroneous, and do another round of QV calculation. But this, since we are having constraints on this, we are just running it 2 times and give the estimate of the first round of long ranger polishing as the QV estimate at the end of the pipeline. Does anyone has questions regarding this? Oops. Are all muted. If you have any questions, feel free to unmute yourself and throw any questions, okay? Okay, I'll keep going. So to be a bit more practical, so this is the latest workflow from yesterday, exist from yesterday. So yeah, so here the input FASTA file will be the one that you get from Arrow, and 10x reads needs to be set in here as a required field option. And when putting the 10x reads in here, just make sure that your read names follow this scheme, which is picked up by Long Ranger. If it doesn't match the scheme, especially this part, I think, please make sure to notify us, either me, Olivier, or Julio, or Marcella. So here, if you don't see these file names in the right convention, you could rename them yourself on DNA Nexus, but we also have to rename them on the GenomeArk. I would suggest to rename them yourself first. And when you go through at the end of the pipeline, you can notify us so we can change the path on the GenomeArk. The reason for changing the GenomeArk at last is when we change the link there, when you change the file name there, the link that's pointing from DNA Nexus to GenomeArk will break. So you will lose the file that you had here. And you have to resync the links, which will take back your assembly process. Okay, there are a few more fields that needs to be set up, I noticed. App requires the genome ID. Here, this is to estimate the NG values of of the assembly at the end. So here you put in the estimates that you got from 10x reads that you got from GenomeScope, or you can put that in that you got originally from Olivier or Eric's estimate that came from other literatures. Excuse me. So at the end, just as usual, get the assembly stats and don't forget to put them back in our track sheet. Make sure that you got the scaffold names properly. I made it automatically to be outputted as T2 and T3. Just make sure that they are in the right positions before you transfer them back. And confirm the final assembly names. So these are the naming convention that we are using for the final assembly that will be sent for curation. Is also picked up by our vgp.github.io. So if you want your genome to be displayed there, make sure you're following this convention here. Rest goes the same, export to GenomeArg. And here again, you can check the QV. Okay, if it's close to 40, but not quite, like 38, 39, because it's the QV of the first round of Lone Ranger freebase polishing. So be sad if your QV seems looking sad, but it's not. It's the lower bound of the QV. And then at the end, notify to our training channel that curation. So here it's an example where the FASTA files should be located when you do the transferring. It's right under the assembly VGP standard something version, the same level as the intermediates folder. So what happens when you notify to the training channel is there will be, we have Julio, Olivia, and Marcella who will be gatekeeping whether these genomes are accessible and are meeting the basic requirements that we set or expectations that we have. So first, check if you can download the final FASTA files. Here, the S3 convention can be automatically transformed to this HTTPS convention, which you can access through wget, or if you just put this line into your browser, you can the download will automatically begin. Use the right species name and the genome ID to the path to the assembly. So these are a few things that our gatekeepers will have a look at the end. I see heat map a requirement, but we'll to check if the continuity looks somewhat weird and seems not being improved after the Hi-C scaffolding. So, do you have any questions up to now?
if I may comment just one
Yeah.
quick thing here. So I would say that probably the QV calculated after the first round of polishing is more accurate in capturing any errors that may have occurred. Assuming that after the first round of polishing, if you're polishing something that has already been polished, then maybe it will not detect that. So Chai has added to the latest version of the workflow a QV calculator app also after the very first round of polishing, and I would encourage to look at that one too.
Oh, you mean?
So in the first place, there was only a final QV calculator app at the very end of the process. Mm-hmm. Now there is another one, so you have also another file that is telling you what the QV is at the intermediate step right after the first polishing. And if there is let's say a disagreement between the arrow polished assembly and the alignment, it will probably be clear there.
Mm-hmm. Yeah, it's a good sanity check to see the QV after the first round of long range polishing. So the QV you get there is the arrow polished assembly, not long range or freebase polished assembly.
Yeah,
just to make sure. Just to let you know that I agree that it's good to check the QV improvements after every time. If you see the QV is not getting any better improved, that means that there could be some happened or silently happened during freebase polishing. Either your long ranger failed or didn't properly polish the variants. I think that's what to say, Julie, right?
Yeah, I mean, if the QV of the first QV is very low, that probably means that there is a mismatch between the arrow polished assembly and the mapped reads from the first round.
Yep, and I have seen a few people making mistakes to put in the pre-arrow polished assembly to long ranger polishing step. So make sure you use T1, the arrow polished assembly. Okay. So I talked. So I sent the curation requests myself before to Kirsten and her group. And now they set up a very convenient system, the Jira system. And Kirsten wanted me to make, including too many people email address in here. But yeah, so if you are not in Singer, I got this email too. You get this email asking about account activation, but you can ignore this. So yeah, anyway, what happens?
Sorry, if I can just comment on this. So this seems to be inherent to this tracking database, and apparently we can't switch it off. Whenever you send an email to Jira at Sanger AC UK for the first time, you get this email back saying we've made you an account, please activate it, and then that doesn't work, so it creates confusion. Whenever you copy or buy copy on this, you will also get that invitation to set up an account and to activate it. So that causes even more confusion. So please make sure that you do not copy anyone when you send this email, the request for curation, and ignore anything that comes back from Jira.
Okay, so in case there are people that in the assembly steps, what would you recommend? To put them in CC or just put them in the foot, the email?
Not at all. Not to Grid Jira, but to everyone else.
Okay. All right. So so the title I put IP in front and the species ID say ready for curation. Since I know Kirsten's group are dealing with a lot of genomes, and just wanted to make them easy to know that this genome comes from the VGP. The genome that we're dealing with is this one. And the species ID already contains the species name, so. Yeah. And in the body of the email, I wrote the paths to the primary and alternate assembly. It could be HTTPS formatted or S3 formatted. I don't think it really matters, since Will and others already are familiar with the AWS S3 CLI, and they download all the raw data themselves for the curation. And then you can include the assembly size and 50s for scaffold context that you got from the end of the pipeline. And you can any additional nodes, paths to the raw data, special treatment, if there were any that happened during the assembly process that you want the curators to know, and the QV, the estimated QV, etc. So here I put some examples of the additional nodes. We had a few genomes that we didn't have Hi-C from the same individual, or didn't have Hi-C at all, or Bionano. Put a note there that this genome without Hi-C or without this platform, or this was generated with Hi-C from a different individual. And for some genomes, you could have low coverage, just to put it in there as a reminder that the QV might be slightly different. And I believe that will slightly change what the regular curation steps. And also contact people if you need, if you know that additional data or special interest might other deeper, such like the sex chromosomes or mitochondria, so on. So from here, I'll leave this slide, or yeah, I will stop share my screen and let Kirsten continue. She will give us an introduction for what will happen during the curation steps.
Can you see my slides?
Mm-hmm.
Okay. I went for a very lengthy title because I just inherited that from the previous talk. But basically, this is all about what we're doing and a bit of why we are doing this. But firstly, I want to say it's nice to be on this call because now I finally see the faces of some people who are sitting in service, so that's great for everyone, and thanks for your service. So when it comes to assembly curation, we tackle this with some experience because 11 years ago we were one of the founding groups of the Genome Reference Consortium, and we have been curating the human reference genome, the mouse reference genome, the zebrafish genome, and also the chicken genome ever since. We've been curating all the genomes that came out of the Vertebrate Genomes project so far, and also additional ones that were produced here at the Sanger Institute. For instance, those that came out of the 25 Genomes project that was started last year for the 25th anniversary of the Sanger, and also additional genomes that were produced on the side of the ordinal assemblies within the Vertebrate Genomes project, and also sort of genomes for friends. Our insights from all this curation was that you probably agree with this, that the perfect data type doesn't exist yet. You need lots of different data types in order to create a decent assembly, and also the perfect algorithm doesn't sadly exist yet. So you need lots of different individual steps in order to get put the data together, and even then at the end, when you've run all your DNA Nexus apps and you're really happy that you have produced an assembly, probably still not in the best shape it could possibly be. And the reason for that is nothing is perfect. Many algorithms are solely trained on human data, so they work very well on human, they work reasonably well on mammals, and then, yeah, weird things happen, and weird things happen in general. So there are always some artifacts that remain or some other issues that still need to be tackled. That's what we are trying to achieve during assembly curation. So there we are looking at the assemblies, and we are correcting and improving the ones that come out of the automated pipeline. And that's mainly by removing contaminations and haplotypic duplications that remained in the assembly despite the haplotype resolution. We then look at whether it's necessary to rearrange scaffolds and contigs because of erroneous fusions or missed joins. Thereby, we try to complete fragmented genes, because after all, that's what everyone wants to see, decent genes. We try to identify and name chromosomal units within the assembly. And all we are doing is based on the GRC's data model and curation. That means we don't just apply any changes because it sort of makes the assembly look good. We need evidence for all the changes. We document the changes so that we can go back what we've done exactly to an assembly. And we do all this by using established file formats. And our main file format is the TPF or TALPath format, which you see here at the bottom right. We basically take your assemblies, we cut them into sizable components and list them in order and orientation. And if we have to make any changes, we can cut those components in even smaller, into even smaller bits, and shove all bits of these files around, rearrange them, fuse them with other bits of files, etc. And then during our post-processing step, we translate this back into FASTA. An obvious side effect of this curation process is that it influences the assembly strategy as a whole. So we can identify shortcomings in current software and applications, and we can advise the teams creating the assemblies, you guys, on what should be part of the pipeline, in which order it should be running, etc., etc. So the assembly curation process starts off with the assembly that you are handing over to us. And it also includes all the sequencing and mapping data that you've been using to create this assembly. So the first thing we are doing is we take this assembly and we run it through decontamination, where we are trying to remove artifacts. We are masking out vector and adapter sequence. We are setting aside organelle genomes like mitochondrial genomes. And we are trying to find out whether any other sequencing data that has no place in this species assembly erroneously went into it. At this point, we might have to feed back and request an adjustment of the assembly because, for instance, it happened before that a wrong SMART cell was included into the Phakin assembly process, and that then gave rise to an assembly that had two different species in the same go, so that should not happen anymore. In this case, the assembly has to be recreated, obviously, but sometimes it's just little adjustments that can improve the situation. After we have a decontaminated assembly, we feed this in GVAL, which is our genome evaluation browser. You see that depicted on the bottom right. It looks a bit like Ensembl because it's based on the Ensembl database, data model, and partly on the Ensembl web code, but it's not gene-centric as Ensembl, but rather sequence-centric. So in there, we load the assembly and we load all the sequencing and mapping data that went into the assembly, and we align everything and we're looking for discrepancies between the data. We are also trawling the internet and use all the additional information that you provided, and that can be any sorts of things. Also into GVAL and look for discrepancies there as well. This then gets packaged up nicely by color coding discrepancies and making this all available to the curators, also in forms of punch lists that list all those discrepancies. And it then goes into a manual curation step where one of our currently 3 curators looks through everything that this assembly has to offer, makes the appropriate changes, and then ends up with hopefully a curated assembly with chromosome assignment that then gets submitted to the DNA archive, so either GenBank or ENA. During the manual curation, obviously, we then have an opportunity to feedback on the assembly and also on the decontamination process. And if we spot anything that looks like a systematic error, for instance, we will make sure to let you know and make recommendations how to avoid that in the future. Maybe I should say here, we had assemblies before that we actually had to send back because they were in not a good enough state to be curated. If I remember correctly, so apart from the one assembly that had the wrong smart cells in it, if I remember correctly, these were all assemblies that had too many haplotype extensions, so duplications created by not removing or resolving the haplotypes correctly. So if we hadn't any assembly back to you guys, don't be disappointed. That won't be the first, and that is a totally normal issue, and yeah, it can happen. So this is what the curators are confronted with. As you can hopefully appreciate here on the left side, you see in blue here. Can you see my mouse? My mouse pointer, by the way. Okay, so here in blue you see the current assembly, and you see aligned here the previous assembly incarnations that were aligned here. And you can see that the initial Falcon unzip assembly was still in two contexts here. They then got joined by the SCAF10X process. So there's a little gap here in between. And exactly at this point, the PacBio coverage dramatically decreases, the BioNano maps don't map anymore, and the synteny breaks. So with close relative breaks. So this would be an indication for us that there is something going on here and that needs to be broken again and rearranged, for instance. We don't, as I already said, we don't just get our information from the browser interface, but also from the punch list, and more or less bespoke for every assembly. So we can list here, for instance, everything that break10x tells us, everything that the BioNano script tells us should be broken or rejoined. We can look at other weird artifacts like zero depth regions from PacBio, extreme depth regions. We look at transcript organization. So for instance, one half of a map or an aligned transcript is here, the other half is there, this should be joined, etc., etc. Here's a brief list of common assembly issues. So the things we are facing on a daily basis and correcting. We sometimes see trailing ends. You can't submit sequence with trailing ends. The DNA archives will complain about this, so we remove them. We sometimes also find extreme gaps. They're usually a relic from running the hybrid scaffolding in BioNano, and then subsequent cutting apart of scaffolds. And this is an example here where you see that basically 80% of this contig or scaffold here is just a gap, and this is something that should not be standing like this, so we modify this. I already told you a bit about contamination. So we look for phage contamination, vector, adapter, everything that can come out of the usual lab procedures. We look for contamination with other species. We set aside, for instance, organelle genomes and inform Julio about them. And based on our detection of incorrect smart cells being joined into the same assembly project, right at the start of the assembly, I believe, are you actually doing this? Are you running the mesh map as part of the DNA Nexus pipeline, or is that even a previous step to this? Run.
Encourage to run mesh map before starting the DNA Nexus pipeline.
So you're all familiar with this, excellent. We see inversions quite frequently. They come out on the Hi-C-map as these little stars of diagonal and indicate that something is not quite right. This is a very nice example where you see that the gene is broken and the right bit of the gene here is inverted, and it's nicely fixable by simply inverting the bit between two contig gaps here. And then what we deal with very, very frequently are both misjoins, which you can easily see in the Hi-C map. For instance, here, this should be two different scaffolds, not a joined scaffold. And then, of course, all the joints that were missed, which you can see here in the off-diagonal red blotches that indicate that this little bit up here actually belongs to this bit and should be joined. The most common error in assemblies that we faced so far, which we can hopefully tackle now within a general assembly step in the future, are duplications that are caused by haplotic retentions. And you can see an example here where you have the same gene represented twice. And this is caused by the region here bracketed in red being there twice. You might appreciate that this is caused by one of the two haplotypes not being removed from the end of a primary FARC and unzip contig here. This is quite a common error, unfortunately, still, and can't be tackled with purge haplotics because purge haplotics only removes whole contigs, not just end of contigs. We hope that in the future purge loops, Dangfang's new program will deal with this. And you can see here that the bionano diverges here because the bionano would match either this going through from here and then continuing there, or alternatively going through from here and skipping this and continuing there. So it's wrong for both alignments in this case because it would obviously only support one of the two haplotypes. Once we've made all those corrections, we hopefully have transformed a genome that has a high CMAP like the one here on the left side into something that looks nice and clean like the one here on the right side. Then we can go and identify those chromosome scale scaffolds that we would like to associate with a karyotype if it's available. If it's not available, at the moment we're using this as an inferred karyotype, so we are assuming that all these chromosome scale scaffolds are representatives of chromosome sequences each, and then we are going forwards and try to name them. We do this either by synteny or by size. So if we have a very close relative available, like in this example here, where we had FGuWill and aligned this with Medaka, which already had a chromosome level assembly of a decent quality, and we can see clearly that each of these chromosomal units or chromosome scale scaffolds aligns clearly to one chromosome in Medaka. We just take the Medaka chromosome numbering so that everyone who does analysis later with does not have to juggle different numbers. If we can't make such an easy assignment, we simply assign names or numbers by size. Very important, obviously, that we have to keep the naming consistent for future releases. So if we should ever re-release an assembly of the same species, we need to make sure that we are taking whatever naming we chose forwards. During this process, we also try to identify sex chromosomes, which you might know is very easy for mammals, as you can see in the graphic here. But then as soon as you leave the mammals, all sorts of weird things happen. So in birds, you have ZZZW systems. And then if you go to fish, all bets are off. It can be all sorts of things. So it's not that easy to spot. And for most fish that we've curated so far, we were actually not able to assign sex chromosomes. But for birds and for mammals, and I believe also for the one reptile that we've done so far, it was actually okay. So for the future, we intend to hopefully also annotate centromeres and telomeres. The structure to do so, to record this information and to submit this information exists. It's not that easy to spot centromeres, but we have access to some lists of likely centromeric sequences for many species, and the more reference genomes we produce, the easier it might become to identify centromeric units. So what we've done so far is we've dealt with over 100 assemblies by now, and that some of them we had to reject. So in total, we have curated about 92 giga base pairs so far. So that comes out as the average assembly size of 1 gig. The average intervention number per 1 gig of assembly is around 150. And you see this broken down into breaks, joins, inversions, and haplotic removals. And you can see that what we do most is removing these little haplotypic duplications that still hang at the end of contigs. And then apparently the assembly process misses out on joins more than it creates false joins, although the false joins have more of an impact on the metrics, on the N50s that you get out of it. And what this leads to, this global creation process, is of course an improved data mapping, because that it was set out to achieve by removing discrepancies between aligned data and the assembly. It improves the gene space completeness measured by BUSCO, although only slightly, because the changes we are applying are on a very high level. So we are not really going in there and changing individual base pairs that might provide a better gene space completeness. But what it definitely does, the whole process, is to change the assembly metrics. But we have to be a bit careful with interpreting the metrics, because what it does in many cases is, as you can see here in the bottom graphics, it might actually make the scaffold N50 sort of inverted commas worse, because there were so many misjoins in the initial assemblies that we had to break, that the final scaffold N50 is much, much smaller. In two cases here, actually halved after the curation process. But that's what the real assembly looks like, so that's all fine. Here are some examples for what assemblies look like before and after curation. So on the top left, you have the same example that I showed earlier, high CMAP of an assembly before curation. At the bottom, the high CMAP after curation, where you can see that everything is now nicely put in chromosome scale scaffolds. On the top right here, you see a comparison between the raw automated assemblies of two different assemblies from the same species. And at the bottom here, you see an alignment of the curated assemblies of these two species, where you can appreciate that they now really look alike, and they are probably much more real than what initially came out of the pipeline. So in summary, we believe that assembly curation improves both the assembly processes and the strategy, and also resolves the remaining assembly shortcomings, and that it allows scaffolding up to chromosome level. And it actually doesn't take that long. We need about a week to produce the GWAS database, and then one curator takes about a week to deal with one assembly. And lastly, I have sort of a mirror image of the last slide that Orang showed about the overall curation process that is sort of visible to you. Please do email gridjira@Sanger SC UK, and the absolute minimum requirement is that you tell us ID of the assembly. Everything else we can probably find out, but you make our life much easier if you provide all the paths to the files, obviously, and you reduce opportunities for confusion here. We already said that please provide any other useful info. Please follow Orang's slide on the instructions and ignore any emails about account setups. We then go and build the database, which you can actually see. It's at vgp-gval.sanger.ac.uk. So in there you can look up your assembly in GVAL if you want to. And we then go ahead, we curate it, we process it, and we submit it to INSTC, and that's the assembly done. And that's all I want to show. Thank you. Any questions?
I have several questions actually, but let's all the training ask the question first. All right, since there's no question yet, if you don't mind, I would like to start with one. So first of all, can you guys hear me?
I can hear you.
All right, sounds good. So first of all, I noticed that there are several fish species that are deployed, and a lot of them are not even full to deploy, more partial to deploy thing.
Yeah.
Do we actually have like any metrics that good for indicate the duplication by the haplotype you mentioned earlier? Because now how we really distinguish between the we can segment out the type, I mean between different haploid from each other, or we don't actually capture all the copy of the polyploid gene.
So far, we've circumvented this problem by creating a sort of reduced polyploid version of these species. For instance, we had brown trout, which was tetraploid and made into a double haploid by a collaborator. So that's much easier to deal with. It remained partially diploid, but we could sort this out, so that was okay. We now are dealing with sterlet, which is tetraploid, so that is actually the first assembly that is not diploid or haploid, or double haploid, that we will be dealing with, and I can tell you more after that. So far we are battling a still very high duplication rate. In Busco, for instance, we were able to reduce this from over 60% to under 30%. We haven't gone through the whole curation process yet, but there are still many lessons to learn with non-diploid species. Yeah, it could be an issue.
I see. Actually, if there is no other question for now, I have one. Sorry. So I think meeting the PAX conference, so talking about that, in the future, maybe we don't have to generate all data type. Right now we have like 10 bio nano and high C. So I heard that there's a strong interest to, let's say, if you want to stick one scaffold, maybe you can try high C, for example. But right now, it seems to me that the bioinformatic tool for this high C scaffold is quite limited. So far I know HiRISE, which the only version that they released was like back on 5 something, and now we have Salsa 2, which a lot more active. Do we have any other, or besides the Salsa 2, which we use currently nowadays, do we have any other tool that we use out there that you have already tried out there that doing quite good job? Concerned that if we come down to just one data type in the future, are we gonna have to just rely on the heat here for the scaffolding, or we have another tool that we can use for cross-validate those kind of thing?
So you're breaking up a bit. Am I right that you ask whether there is an alternative for Salsa for high C scaffolding?
Yes, that's correct.
So so far we have only tried Salsa, but we have more than 20 genomes that are scaffolded by Eris Lieberman group, who uses JUSA, JUICEBOX, etc. The whole 3D mapping, and we hope that we get some data from them soon, and then we can compare to Salsa.
What's the name again?
Sorry.
What's the name of the tool again?
Eris Lieberman group, so the inventor of high C, if I am correctly informed.
3D DNA.
The
DNA.
Yeah. Okay.
So they have this JUSA and JUICEBOX
that's okay.
Yeah. So for lining data and for curating high C data, we initially used JUICEBOX, but the resolution was really bad, and the curation support in JUICEBOX just made all our machines crash. So our experience with that is limited. We are using HiGlass in order to view the high C maps, and Ed in the Min Ning's group has recently created some software called Pretext, which I personally find really excellent because it's fast, very slim, and allows shoving data around. So you can actually apply whatever you want to change to your assembly in there. So that might be a nice contender for looking at high C maps in the future.
I see. But I see. I think I got it.
So if I may, I may have a more general comment and question. So because Christian, you said that I'm thinking about the short-term goal of this summer. So it's I think you said last time in the VGP call that you were thinking about having like 4 or 5 genomes curated per week. Is that correct? Okay, and so I assume you should have them every week, like sort of at least, so that you can start, let's say, on Monday and and move through. So I'm wondering whether you also have, I mean, you, Arang, and probably everybody in the school is involved. It will be involved in the August goal, isn't it? So I'm wondering what do you think about how to move forward, because now we have, I think we have been sending out for polishing most of the genomes for curation, most of the genomes that were in the polishing or in the later stages of the pipeline. And so we probably now should start over with the new ones, but I'm not sure about the stage in which most genomes are. So
you can't really overload us. We just work it all off as it comes. We might have 50 queuing and then we don't feel so well, but yeah, we can just do what we can do. So please don't hold back on anything for our sake. Produce as many as you can and we just pick them up as we go along.
Okay.
We'd rather have a totally full queue than as we did over the past 2 weeks, sort of wait for things to drop in. We obviously also get some assemblies from Shane here, and the Darwin assemblies are coming now, competing with the VGP assemblies. So yeah, just keep them coming. What's there will be picked up at some point. But it's holiday time now, so everything slows down to a crawl. No one's here.
Hey, can I also make a question?
Sure. Go ahead.
Thanks, Arang and Kirsten, for the talk. It was really good. I think everybody really benefited from the call. Kirsten, what do you think? Why do you think BioNano is inserting these very large gaps?
It's to reflect the real situation in the genome. So the big gap might actually be justified, and there might be a scaffold flying around somewhere that fits into that gap.
Okay. Okay, thank you.
So Arang, do you have any comments on how to move forward from now on, or because I think for now there may be like 3 more genomes that could be sent out for curation, but I'm not sure what's.
I think we should send them as soon as they are ready for curation and start those genomes that are ready for beginning the pipeline.
Yeah, no, that's my point. So that probably most people that were in charge of one genome or 2, they have completed the pipeline. So maybe we should assign more genomes, or I don't know. What do you think about it?
Yeah. Yeah. To be honest, I haven't checked which genomes are ready for the pipeline.
Yeah.
Yes. Yeah. Yes. Yeah. Maybe Olivier. I think you are you are dealing with most of the sequencing data. Can you set something like data ready or something in the project tracking sheet? So,
okay, I'll do that.
Yeah. And yeah, I guess people are still willing to contribute and volunteer to the assembly pipeline or the assembly process. Feel free to ping us on the training channel.
Yeah, in a lot of cases we don't have all the full data sets like PacBio and 10x. Just say it's ready to start, and people can start on that.
Yeah.
Even if you have only PacBio, I would start.
Okay, I will flag them to be okay.
On that note, I would like to introduce a new student, the volunteer to assemble genomes. This is Thomas. He's just starting a PhD student from Camilla here in Berlin. And we have data for the green turtle that is coming from Dresden, and we are assembling it in our servers locally. So I am supervising Thomas in running everything locally too for the green turtle.
I see. That's great.
And one more comment is also I'm assembling another memo, which is Toad. And I assemble the Falcon, and then I just put it through Purge dupes. I am running the K-Mayor analysis now in comparison with the reads with the assembled, and we can have a look, but I guess it looks good. And we have to discuss that, right? Because I think the 1.6 pipeline would put the Purge dupes in the, just after Falcon, I guess.
Yeah, Purge dupes is in the plan. I pushed that into the priority list try, but seems like there were some issues with the Falcon app and the Salsa app, but I think it's now under high priority. So there will be I guess we have to change the pipeline name to something like version 1.6 or 1.7, just to make it consistent that we are using Purge dupes.
Sure.
So your guys' impression with Purge is quite good, right? I mean, better than Purge Hypothesis.
What was your question, Chai? Sorry.
So far your impression on Purge is better than the Purge Hypothesis, or in most species that you all try, right?
So the Purge dupe assemblies that we've seen so far look very good. One of the main statistics is that in the filling step, right towards the entity assembly process, we actually fill an awful lot of gaps. There's now contigs that sit next to each other, belong next to each other, and there are duplications in those neighboring contigs. We've also seen, so the last assembly we looked at, we had to not remove nearly 100 haplotic retentions, but 2. So it's really on a different scale. Yeah.
That's all very good.
Yeah, seems like that will help the curation step a lot.
Oh yeah.
I have one more question on the contamination that Kristin mentioned. So I think right now for the DNSS pipeline we don't have the match checking yet, but we already have the applet actually, so we can share with all the training very soon. But then how should we communicate with that result with the team though? Let's say after I give to all the trainee and then trainee run the the match disk, get a heat map thing here, and like what the process after that, or like even if after we found that this data is contaminated, should we ping someone to clean it there? Is that what the process should look like?
Ping me.
Okay. All right, sounds good. And basically, did you just try to like run on this data set, or like are you running blast on all the raw data?
Yeah, we've done that. Yeah, that's the process.
Okay.
And Julio, what he was doing too is you can assemble the mitochondria and double check.
Okay, all right, that sounds good.
Yeah.
So I'll put the applet in the project, so can run that as well. Okay.
Would be good to have an app that automatically runs the contamination checking. So yeah, I think in the plan we have the Purge dupes that's gonna be updating our whole assembly pipeline. And Julio's mitochondria assembly pipeline becomes automatic process. We can stick that in before the aero polishing, so we are not over polishing the numpties that are in the.
So with respect to this, I sent an email to Kirsten yesterday. So so far I have the pipeline ready and it works on Sanger, and I'm about to set it up also here at the Rockefeller because they just granted me access to the cluster. And one thing here is that we could even start immediately because as soon as we have the arrow polished config from the Mito, then of course it doesn't necessarily have to be already the perfect Mito assemble. If you just add that particular config to your assembly, that is subtract all the miter reads anyway. So I think, as you asked me to modify SALSA, I will look into that, and if it's manageable, I think we can start that process right now, even without having the full pipeline established already.
So you will use a Mito assembly? Sorry, go ahead, question.
I want to say, I'm sorry, I haven't dealt with your email yet, Julio.
Okay, no problem.
But what I would like to tell you is that, so we've just dealt with Otter here, and Otter had a blood parasite. The blood parasite had an apicocluster. So that is a different organelle genome. So I wondered how your pipeline will deal with this if you don't just find the mitochondrial genome of the species you want to sequence, but also mitochondrial genomes of parasites or apicoclustids.
No. Yeah. Okay. So the pipeline in the first place was meant to assemble the supposed mito for that particular species. So it's not meant to assemble any mito that may be there, even if you don't expect it to be there. In order to do that, I think we have to follow a different approach. We have been doing something similar. I mean, Olivier has been doing something very similar recently, to just check for contamination and that sort of things. We can probably rely on having a data set of a sort of curated data set of all the mitos that have ever been spotted at some point, and we can say, okay, let's try to see if there is any mito rather than the mito I'm looking for. So that's something we can look into.
I mean, we should be able to identify these things during our decontamination, because it takes into account everything that was ever submitted. So it should hopefully find that. But of course, that is sort of too late for your pipeline. That's why I'm asking.
No. So the thing is that for now, the pipeline is using the reference of let's say the same species or a closely related species for the first round of assembly, under the assumption that we're looking for that particular mito, not for any mito. But if you instead use a, let's say, a very large reference data set with any possible mito, of course, in the case there are other mitos present that you are not aware of, but they are still in the gene bank. And if Canu is able to assemble them independently, which should be the case if the contamination is high enough, then you will detect them in the subsequent phases. So that will work. Of course, it increases the runtime, because then you have to map everything to a much larger, let's say, reference data set. But it's something. It's also. I mean, it will also be the most appropriate way to look for cross contamination, or as you said, parasites or whatever else.
The mapping time is not that bad. I mean, I tried on a data set with 700 mitochondrial genomes, and you map PacBio data very fast.
Yeah, I mean, they are small, so it should work anyway. So.
One final comment. It's simple, but the trimming of the R1 for the 10x read the weekend chimera composition would be nice. Because just to trim the adapter, sometimes I have to download all of the data. So if this can be in the DNA Nexus too, it would be good.
Yeah, I have to make the trimming asset for on the Nexus platform as well.
Thank you.
So you use the trimming that's used in SCAF10X. Try just as a note. Follow up on that slide. Okay, do we have any more questions or comments? Ready to receive a new genome?
So, but what do you think about my proposal of having a more consistent a drop-down menu with all the steps? So
I agree.
Okay.
I like that idea.
Oh, so I will do that.
Yep.
I love especially the number, make it clear. That's good.
Mhm.
Okay. Cool.
All right. Thank you, everyone. Especially thank Chai and Julio, Olivier, Marcella, Kirsty.
Everyone actually appreciate everyone joined the call and especially around and prepare to talk today. Thank you so much.
Thank you, everyone.
All right. See you, I guess, on Friday.
So,
Yeah.
Bye, guys. See you.
Bye.